Okay, thank you very much, everybody, for your patience. Bismillah ar-Rahman ar-Rahim. I am Rouhollah Fathi, the Associate Professor of Embryology Department in Rouyang Institute. And at first, I have to thanks to my dear colleagues in Eastern Mediterranean University, special Professor Tasman, Professor Alios Turan, and Professor Vaziri for organizing this mutual meeting between Rouyang Institute and Eastern Mediterranean University. I hope that this the first mutual program between Rouyang and EMU leads to continue our collaboration in the future for education, research, and every scientific events. Okay. Okay. At first, let me to remember our dear late founder of Rouyang Institute, Professor Kazemi Ashtiani, who established Rouyang Institute over 30 years ago. My speaking about the is about the cryobiology, but the cryo injuries after the cryopreservation. Dr. Zainab had a good statement about the cryopreservation and its different aspects of cryopreservation and its indications. But I want to share with you about the cryo injuries, the main challenges in cryopreservation. And you know that I have to say that cold or cryo isn't a real thing. It isn't a real subject. And when we take the temperature from everything, it goes to cold and to cryo. So in the less of temperature, you can see the coldness. But after cryopreservation, after cell and tissue cryopreservation, especially after sexual cells and reproductive tissues cryopreservation, we are worried about the cryo injuries. That when cells and tissues go underwent the very low temperature until minus 196 centigrade, it is a very high stress on the cell and tissues. And we are worried about the damages on the cells and tissues. Dr. Zainab spoke about the two main different protocols for cryopreservation. The first, let me to activate my laser pointer. Okay, special slow cooling or slow freezing and rapid cooling or vitrification. And you know that in slow cooling we less the temperature gradually, but in rapid cooling or vitrification we immerse the samples immediately in liquid nitrogen with a temperature about near to minus 200 centigrade. So in slow cooling, the different materials with different temperature for freezing, they experience their cool freezing temperature, and every elements have a fracture between each other in different temperature during the freezing. But in vitrification or rapid cooling, when we put the sample in liquid nitrogen, they experience immediately the temperature near to minus 200 centigrade, and it is very high, very low temperature. So different materials don't touch the freezing point, and they fixed immediately all of them. So the cytoplasm of the cell and any materials in the tissue go to a similar situation like vitreous. So we call this method vitrification. So in vitrification, we eliminate the ice crystallization, but in slow cooling, we experience and we can see the ice crystallization in the cell. Also, you know that the water crystal after freezing, it has many very tips that can damage to the cytoplasmic organelles or cell membrane and nuclear elements in the cell. For preventing the cryo injuries, we use the cryoprotectant that this material can prevent the cells and tissue, protect the cells and tissues from the different damages due to the cryopreservation and freezing. And the other main cryo injury is the osmotic pressure or osmotic shock. You know that when we put the cells and tissues in cryoprotectants, the water and cryoprotectant will be changed with each other, and the water goes out of the cell and the cryoprotectant goes into the cells. And it should be changed very fast. But sometimes when water is going out of the cell, is going to go out of the cells, cryoprotectants enter very fast, and cells go to swelling, and it can experience the swelling. And in the other side, when water goes out of the cell very fast and cryoprotectant cannot enter to the cells goes to the shrinkage, and the cytoplasmic organelles and nuclear materials can damage and fracture. And so we can cryoprotectants to prevent the ice crystallization, no? But ice crystallization, I mean that the cryoprotectant, they have another damage to the cells, that they are toxic. When in Royal Institute, we have a good experience for freezing the sexual cells like sperm, oocyte, and embryos. And for sperm cryopreservation, we can see one of the main cryo injury as DNA fragmentation. Also, when the sperm DNA goes to fragmentation, sperm doesn't work truly, and it cannot fertilize the oocyte. And this is the main cryo injury during the sperm cryopreservation. And so DNA fragmentation leads to apoptosis and maybe mutations. And maybe some sperms within the oocyte after fertilization can repair themselves, but many of them cannot. For oocyte cryopreservation, we are worried about the post-cryopreservation cryoinjuries, like reactive oxygen species, alteration in gene expression profile of the oocyte, and reduction of mitochondrial activity, and aneuploidy, incidence of the aneuploidy, and hardening of the zona pellucida, which is the authority membrane of the oocyte and embryo, and it protects the embryo until the implantation, when the embryo wants to implant in the uterus. And toxicity of cryoprotectants, wrinkles, and damage to organelles, and IVM rate decrease. And you know that after cryopreservation for immature oocytes or germinal vesicles, we can see the reduction of IVF rate or in vitro fertilization rate in the oocyte. And for embryo cryopreservation, we are worried about the reduction of IVF rate and dysfunction of the ZGA or zygote genome activation. You know that ZGA will be happen in mouse in 2-cell stage embryo, and for human it will be happen between the 4 and 8-cell stage embryo. So it is very important. If embryo cannot start the ZGA, it leads to arrest, and it doesn't reach to the implantation point. And decrease rate of transformation in morula to blastocyst stage, induction of epigenetic changes, and increased repair genes expression, and decreased mitochondrial function. And for embryo, one of the very, very important thing is the reduction of implantation ability. Okay, so this is our last publication for oocyte vitrification, and we evaluated the capability of the oocyte after vitrification for starting the repair gene expression in sperm with high level of DNA fragmentation. And so we saw that after oocyte vitrification, the vitrified oocytes miss its capability for repair the sperm, especially in the situation of high level of DNA fragmentation. And what is the solutions? Difference, we involved with different solutions for reduction of the damages after cryopreservation. We can use many material after cryopreservation. We call them antifreeze molecules, like the heat shock proteins, antifreeze proteins, magnetic field. It is different from the molecules I explained. And cryoprotectants, the chemical cryoprotectants like glycerol, dimethyl sulfoxide, ethylene glycol, and natural cryoprotectants like the honey, and some saccharides like xylomonan. And special, we used the nanoparticles and nanocomposite, and especially the magnetic field. Okay, you can see here we set up a new terminology in cryopreservation. We call it ACT, assisted cryobiology techniques. We used the nanoparticles and nanocomposites for prevent the toxicity of the cryoprotectants, and a static magnetic field for change in the speed of exchange between the water and cryoprotectants during the dehydration and rehydration in cryopreservation. Okay, you can see here our last publications in the below of this table that we use the static magnetic field for mouse cryopreservation of mouse ovary, SMF for cryopreservation of mouse oocytes, and nanoparticles, special Fe3O4 for the vitrification of mouse immature oocytes that we call them germinal vesicle, and also integration of the static magnetic field and nanoparticles for cryopreservation of the mouse oocytes. Okay, this slide is very interesting for me, and I think that for the other researchers who are working in cryobiology field, especially for cryopreservation of the sexual cells. After freezing vitrification of the oocytes, we saw many vacuoles in the cytoplasm of the oocyte. This is the ultra-structure assessments of oocyte. We call this technique TEM, TEM, transmission electron microscopy. Okay, after vitrification without magnetic field, you can see many vacuoles in the oocyte cytoplasm. But after using the SMF, a static magnetic field, during the vitrification, you can see there is no vacuoles, or it is very less vacuole in the oocyte cytoplasm. And this in this graph you can see this difference that is the meaningfully and statistically significant. Okay, this is another picture for our another work after using the static magnetic field and integrated with nanoparticles, Fe3O4. And you can see beside of the protection of cryo injuries in vacuoles, you can see that the other structures like the microvillus in the surface of the oocyte, it protects during the vitrification in integration of a static magnetic field and nanoparticles. So what's happened during the application of nanoparticles? Nanoparticles can enhance heat transfer, inhibit ice crystallization, prevent DNA damage, and prevents oxidative stress, and reduce the RAS production, and structure protection, spatial ultra structure protection. And you can see very nice illustration and very nice picture from the nanocomposites of Fe3O4 nanoparticle and honey that we green synthesize this nanocomposite and we use it for our experiments. And this is the high magnification of these nanoparticles. It is a very nice picture of the nanocomposite of Fe3O4 and honey. After vitrification, you can see the survival rate. It is less than near to 60%. But after using the nanocomposite, Fe3O4 and honey nanocomposite, you can see it's higher than the vitrification, and this difference is statistically significant. And after cleavage of the embryo and after in vitro culture of the embryo, after fertilization of the oocytes derived from vitrification using of the nanocomposite of Fe3O4 and honey, we culture the embryos until blastocyst formation. And you can see here the green column has a very statistically significant difference from the vitrified without any nanocomposite. And you can see meaningfully different, and it's near to the fresh embryos. And it is a very interesting result for us. And you can see very nice picture that my students send it for me. It is the hatching embryo after vitrification with nanocomposite of honey and epitropha. But in Rouyang Institute, we have many cryobiology activities, and we have a very powerful group in cryobioengineering. And we have an annually symposium, and it will be held in each April. It's the 9th Cryobiology Symposium coming in the 2026 year. And we invited the foreigners and the scientists from the other countries. He is the Professor Jason Acker, the previous head of Society, World Society of Cryobiology, who came to Iran and is visiting the, you can see that he is visiting the Rouyang Institute. And Professor Luciana Dini from the Italy, Rome University, Sapienza University, and we have a very nice school, joint school, cryo school, and I hope that one day we can present this cryo school in Eastern Mediterranean University. And many international webinars and very nice webinar that Rouyang GEN, Rouyang Global Education Network, and we can set up these webinars with EMU, inshallah, in the future. But my last presentation is about a very interesting Rouyang program, a scientific program for school students. And we have annually create a school for school students. You can see here that they present in our program. They were over 400 students, and we held for them a cryopreservation of the many materials on the bench, and they touch these these events, and it was very interesting for them. And now let me to show one very movies. I hope that you can see, you can watch this movie. Okay, and you can see my dear students and colleagues in the Cryobioengineering group in Rouyang Institute, and it is another picture in the Iranian Teacher Day, and with my colleagues and my dear colleagues and dear students. And I hope that one day the EMU scientists and researchers and students will come to Iran and visit the Tehran city, especially in winter, with this very nice view. Okay, thank you very much for your attention.